Mycology is a branch of biology that is concerned with the study of fungi. Whereas medical mycology is the study of mycoses. Mycoses refers to infections that are caused by fungi. The fungi are a group of eukaryotic, heterotrophic, and achlorophyllous microorganisms. They are essentially aerobic, with limited anaerobic capabilities and can reproduce sexually or asexually. There are approximately 80,000 species of fungi. And 400 are medically important. Luckily, there are less than 50 species of fungi that are responsible for more than 90% of the fungal infections of humans and other animals. They are capable of causing superficial, cutaneous, subcutaneous, or systemic disease. And most of these pathogenic fungi are exogenous. Fungi seen in the clinical laboratory can generally be separated into two groups based on the appearance of the colonies formed. The yeasts produce moist, creamy, opaque or pasty colonies on media. Whereas the filamentous fungi or molds produce fluffy, cottony, woolly or powdery colonies. There are also several pathogenic fungi that are dimorphic. Dimorphic fungi are fungi that can exist in the form of both mold and yeast. This is usually brought about by change in temperature, and the fungi are also described as thermally dimorphic fungi. An example is Talaromyces marnefae, a human pathogen that grows as a mold at room temperature, and as a yeast at human body temperature. Medically important thermally dimorphic fungi includes the following: Histoplasma capsulatum, Blastomyces dermatitidis, Paracoccidioides brasiliensis, Sporothrix schenkii, and Penicillium marnefae. Coccidioides immitis is a dimorphic fungi, but its forms are not dependent on temperature changes. And lastly, you have the Congida species. They are polymorphic, meaning they have yeast, pseudohyphae, and true hyphae forms. Similar to coccidioides, its forms are also not dependent on temperature changes. Yeast are unicellular organisms that are round to oval and range in size from 2 to 60 micrometer. The microscopic morphologic features have limited utility in helping differentiate or identify these organisms. The differentiation of yeasts in direct microscopic and histopathologic examination of clinical specimens is often impossible. But, sometimes, particular characteristics are seen that suggest identification or are pathognomonic. Important morphologic characteristics that are useful in differentiating yeasts include the size of the yeast, the presence or absence of a capsule, and a broad-based versus narrow-necked budding. This photomicrograph depicts the yeast form of Cryptococcus neoformans using a light India ink staining preparation. Life-threatening infections caused by the encapsulated fungal pathogen, Cryptococcus neoformans, have been increasing steadily over the past 10 years because of the onset of AIDS and the expanded use of immunosuppressive drugs. In general, the yeasts reproduce asexually by blastoconidia formation or budding, and sexually by production of ascospores, or basidiospores. The process of budding begins with weakening, and subsequent outpouching of the yeast cell wall. This continues until the bud or daughter cell is completely formed. The cytoplasm of the bud is contiguous with the cytoplasm of the original cell. Finally, a cell wall septum is created between mother and daughter yeast cells. The daughter cell often eventually detaches from the mother cell. The basic structural unit of the molds are tube-like projections called hypha. The hypha grows and elongates at the tip. As it grows, it becomes intertwined to form a loose network. This loose network or mass of hyphae is called a mycelium. Some references would note the fungal thallus, or the body of the fungus, and mycelium are synonymous. Hyphae, from the Greek word huphie, means web. It is a tubular, transparent filament, and usually branching. It is composed of an outer cell wall and a cavity lined or filled with protoplasm including cytoplasm. The major structural polymer in fungal cell walls is typically chitin, in contrast to plants and oomycetes that have cellulosic cell walls. Based on the presence of septa, the hypha can be classified as a septate hypha, or non-septate hypha. Some fungi have hyphae divided into cellular compartments by walls called septa. Septa have tiny perforations which allow molecules, cytoplasm, and sometimes organelles to move between the cells. Fungi can close their septa if they are injured, preventing fluid loss from the rest of the filament. There are many species of fungi with septate hyphae, including those in the genus Aspergillus, and the classes Basidiomycetes and Ascomycetes. When Basidiomycetes mate with each other, the septa of one of the parents degrades to allow the incoming nuclei from the other parent to pass through the hyphae. After the nuclei have established themselves, the septa are reformed. In some species of fungi that have wide hyphae, the septa act as support structures in addition to being barriers. When hyphae grow at their tips, the septum does not form right away. As the cell matures, the wall grows out into the cytoplasm, eventually spanning the width of the hyphae. Non-septate types of hyphae are also called aseptate, or cenocytic. They represent a more primitive form of fungi, and are the ancient ancestors of septate hyphae. Fungi of the genus Mucor, and the division Zygomycetes are non-septate. Non-septate hyphae do have some septa, but they are found only at the branching points. If there were no septa at all, the entire fungus would be at risk of compromise if even one hypha were damaged. Non-septate hyphae are the result of the nucleus repeatedly dividing but not the cytoplasm. This can result in many nuclei in the cytoplasm along with other organelles such as ribosomes, endoplasmic reticulum, and Golgi apparatus. As mentioned in the prior slide, hyphae can be classified based on the presence of internal septa. Hyphae can also be distinguished from species which produce pseudohyphae. Pseudohyphae is a form of incomplete cell division, in which the dividing cells do not separate. There are several yeast species which produce such pseudohyphae. Most yeast reproduce by budding, which is initiated by a lateral or terminal protrusion of new cell growth that enlarges during mitosis. They usually form a septum then separates from the parent cell. But some species produces buds that fail to detach and becomes elongated. The mycelium has two components: the aerial mycelia and the vegetative mycelia. The vegetative mycelium is also known as the substrate mycelium. It is the part of the mycelium grows into the substrate and absorbs nutrients. The substrate mycelium inevitably develops from the germinating spores but never produces spores itself. The aerial mycelium is more closely packed and appears darker than the substrate mycelium. It is the portion of the mycelium above the surface of the substrate. The aerial mycelium also bears the reproductive portion. The term, spore, is derived from the Greek word, spora, which means seed. It is also related to the Greek word, sporos, which means to sow. Spore is a unit of sexual or asexual reproduction that may be adapted for dispersal, and for survival, often for extended periods of time, and in unfavorable conditions. Depending on the fungus, some species can entirely reproduce asexually, while many other species are also capable to reproduce both sexually and asexually. This diagram shows a typical reproductive or life cycle of a fungi that can reproduce sexually and asexually. Let's first look at the asexual reproduction of the fungi, starting with the mycelium. Mycelium is a network of fungal threads or hyphae. These are haploid multicellular structures that produces spores through mitosis. These spores, on a suitable substrate, germinates to produce one or more filamentous processes called germ tubes. Each germ tube elongates to form a hypha. The sexual reproductive cycle of the fungi begins when mycelia from two different spores meet. The protoplasm of two parent cells fuse in the process called plasmogamy. This stage is called the heterokaryotic stage. It means that the organism having genetically different nuclei at the same cell. This is followed by fusion of the nuclei called karyogamy. At this point, you would have a zygote that usually grows to form your fruit body. The fruit body then produces haploid spores through meiosis. And the cycle continues. The increase in number of opportunistic fungal infections in humans is due in large part to the immunocompromised nature of the host. However, there are certain factors, virulence factors, which make it easier for them to invade tissues and cause disease. Some virulence factors have been known for years and include: the first factor is the size of an organism. Example: disease that begins by inhalation requires the organism to be small enough to reach the alveoli. The second factor is the ability of an organism to grow at 37 degrees Celsius at a neutral pH. The third factor, conversion of the dimorphic fungi from the mycelial form to the corresponding yeast or spherule form within the host. And the last is possibly toxin production. The taxonomy of fungi is in a state of constant flux, especially due to research based on DNA comparisons. Current classifications are based on phylogenetic analysis and genetic systematics, which have largely replaced the older classifications. Phylogenetic analysis provides an in-depth understanding of how species evolve through genetic changes. Using phylogenetics, scientists can evaluate the path that connects a present-day organism with its ancestral origin, as well as predicting the genetic divergence that may occur in the future. Whereas molecular systematics is the use of molecular genetics to study the evolution of relationships among individuals and species. To be formally recognized by taxonomists, an organism must be described in accordance with internationally accepted rules and given a Latin binomial. The rules that control the bionomenclature are very diverse and depend on the type of organism. The nomenclature of fungi, including yeasts, is governed by the International Code of Botanical Nomenclature, as adopted by each International Botanical Congress. Any proposed changes to the code are published in Taxon, the official journal of the International Association for Plant Taxonomy, and then debated in the Congress for approval. The kingdom fungi have 8 recognized true divisions of fungi. These are the Chytridiomycota, the Zygomycota, the Ascomycota, the Basidiomycota, the Glomeromycota, the Microsporidia, the Blastocladiomycota, and lastly, the Neocalamastogomycota. An older classification scheme grouped fungi that strictly use asexual reproduction into Deuteromycota, a group that is no longer in use. Note: Mycota is used to designate a phylum, or division, while Mycetes formally denotes a class or is used informally to refer to all members of the phylum. The botanic taxonomic schema for grouping the fungi has little value in clinical microbiology. And thus, clinicians find value in categorizing fungi into 5 categories. The clinical nomenclatures used for the mycoses are based on the 1. Site of the infection, 2. Route of acquisition of the pathogen, and 3. Type of virulence exhibited by the fungus. The classifications are: 1. Superficial mycoses. 2. Cutaneous mycoses. 3. Subcutaneous mycoses. 4. Systemic mycoses. And the last category, the opportunistic mycoses. Clinical specimens collected for microscopy and culture are determined by the site of infection and condition of the patient. When collecting specimens, all efforts should be made to collect specimens for fungal culture as free from bacterial contamination as possible. It is therefore important that all specimen be obtained using aseptic technique, and it should be transported to the laboratory within 2 hours. The scalps of patients with suspected tinea capitis may be examined with a Wood's lamp. Infectious organism would glow or illuminate under the lamp for easier examination and collection. When collecting, fluorescent distorted or fractured hairs should be removed with forceps. Infected hairs can easily be removed, but normal hairs are more difficult to dislodge. A comb or brush may be used to collect loose hair and skin squames. Skin, when involved, should be cleansed with an alcohol wipe before a specimen is collected. Epidermal scales at the active border of a lesion should be removed with a scalpel. Nails should be cleansed with alcohol wipe, and the outermost layer should then be removed by scraping with a scalpel. Deeper scrapings, debris from under the edges of the infected nails, and nail clippings from infected areas are also suitable for culture. Samples of hair, skin, and nails should be collected and placed in a sterile culture dish for transport to the laboratory. Storage at 4 degrees Celsius is not recommended, since at least one dermatophyte is susceptible to cold temperatures. In addition, storage in closed containers is unsuitable due to overgrowth of contaminating bacteria and saprobic fungi in a moist environment. Nail clippings may be grounded in a mortar before being inoculated onto culture media. Skin scrapings and hair may be inoculated directly onto the surface of appropriate culture media. All body fluids are collected aseptically by needle aspiration and should be sent to the laboratory in a sterile container as quickly as possible. However, if a delay is unavoidable, CSF should not be refrigerated, since it is an excellent culture medium and fungi will continue to replicate at 25 to 30 degrees Celsius. Other body fluid specimens may be stored at 4 degrees Celsius overnight, if necessary before culturing. All body fluid should be concentrated by centrifugation for 15 minutes, and a minimum of half a milliliter of sediment should be inoculated onto the surface of media. Small volumes of CSF not suitable for centrifugation may be dropped directly on media surface. As with the collection of other sterile body fluids, good skin antisepsis should be practiced for blood sample collection. 10 milliliters of blood should be collected at periodic intervals as determined by the physician. Blood is inoculated to blood culture bottles at bedside. Bottles should be returned immediately to laboratory for incubation at 35 degrees Celsius for 28 days. Bone marrow aspirates and biopsies are collected after good skin antisepsis and are commonly submitted to the laboratory in a sterile syringe or tube containing EDTA. The collected specimen should be transported to the laboratory as soon as possible. However, it may be refrigerated for no longer than 12 hours, if immediate culturing is impossible. Direct plating on mycology media can be done at bedside. Media may be obtained from the microbiology department. The urinary meatus must be adequately cleansed if a clean catch or catheterized specimen is to be submitted for culture. Suprapubic aspirates are obtained after good skin antisepsis is used in the area of aspiration. Specimens should be cultured promptly, since bacteria and yeasts replicate rapidly in specimens kept at room temperature. If specimens cannot be cultured soon after receipt, they should be refrigerated at 4 degrees Celsius for no longer than 12 to 15 hours. 24-hour specimens, or those collected from indwelling catheter collection bags, are not suitable for culture. Vaginal and cervical specimens collected by a physician are usually submitted on a swab. Transport to the laboratory should be rapid, however, overnight refrigeration before culturing is satisfactory. Specimens should not be stored at room temperature. Specimens from the ear, nose, nasopharynx, and mouth are usually submitted on a sterile swab. Whereas, specimens from the lower respiratory tract should be collected in a sterile wide-mouth bottle or sputum cup. A first morning expectorated sputum specimen is optimal. Before a sputum specimen is collected, the patient's teeth must be extensively brushed or his or her dentures be removed. The mouth should be cleansed by a mouthwash or several rinses of sterile water of saline. For those patients incapable of expectoration, sputum induction may be necessary. Specimens should be transported to the laboratory as soon as possible to ensure maximum recovery of fungi. If culturing is delayed, specimens may be refrigerated at 4 degrees Celsius.
Hi, I'm Dr. Richard Usatine, Associate Editor of the Journal of Family Practice and column editor of Photo Rounds. I am a professor of family medicine, dermatology, and cutaneous surgery at the University of Texas Health Science Center in San Antonio. I am also the medical director of the University Skin Clinic. Today we will demonstrate the KOH preparation for diagnosing fungal skin infections. The quickest and most accurate method for diagnosing fungal skin infections is the KOH preparation. While the fungal culture is more sensitive and specific, it can take days to weeks before the diagnosis is known. The first step is to scrape skin from the affected area onto a microscope slide. This can be performed with the edge of a slide, a scalpel, or a curette. Scrape the edge of the lesion to maximize the yield. A sharp skin curette is a very useful tool for scraping subungual keratosis or nails looking for onychomycosis. A cover slip may be used to move the scrapings into the center of the slide before applying KOH. In this case, a fungal stain with KOH is being used. 2 drops of the Schwartz-Lambkin stain will stain the fungal elements blue and begin to dissolve the human cells. A cover slip is placed on the slide, and the slide is heated briefly with a flame. Heating with a flame is optional, but often speeds up the process of human cell dissolution, making it faster and easier to find the fungal elements. Examination of the slide starts at 10 power. The clinician looks for clusters of cells with associated fungal elements such as hyphae or pseudohyphae. While looking for fungal elements, it is important to use the fine focus to search up and down through the specimen, keeping in mind that the preparation is three-dimensional and hyphae may be seen better at one level than another. Once structures suspicious for fungal elements are identified, the objective is switched to 40 power to confirm or dispel the diagnosis. The most common causes of false positives occur when the clinician overreads overlapping cell borders or other artifacts as hyphae, or overreads micelles from topical medicines as yeast forms. The goal is to find fungal elements on high power with definitive fungal morphology, such as septate branching hyphae or pseudohyphae with buds. False negatives occur when the scraping is inadequate or the clinician does not look at sufficient low-power fields to screen the slide. See how the fungal stain helps the fungal elements stand out from the human cells and therefore increases the sensitivity of the preparation. These slides at high power show other examples of positive KOH preparations for dermatophyte fungus using Schwartz-Lambkin stain. This technique can also be used for Tinea versicolor, with the result showing Malassezia furfur with a ZD and meatball pattern. It is often difficult to tell the difference between a fungal skin infection, an eczematous eruption, and psoriasis. A properly performed KOH preparation is a powerful tool in the diagnosis of skin diseases.
In formal and fixed biopsy specimens, fungi can be detected with the routine hematoxylin and eosin histopathological stain. However, specialized fungal cell wall stains are more sensitive. The two most common stains are Gummery methenamine silver and periodic acid Schiff, which stain fungal walls black or red, respectively. In most cases, the culture is more sensitive than the direct examination, and a portion of the material collected for microscopy should be cultured. There are two general types of culture media used to ensure recovery of clinically significant fungi from clinical specimens. One medium should be non-selective, such as brain-heart infusion agar, which will permit the growth of virtually all clinically relevant fungi. The other type of media should be selective, specially tailored to isolate specific pathogenic fungi of interest. For optimal recovery, a battery of media should be used, and the following are recommended. 1. Media with and without cyclohexamide. Cyclohexamide is added to inhibit the growth of rapidly growing contaminating molds. 2. Media with and without an antimicrobial agent. Antibacterial agents are used to kill the contaminating bacterial species. Chloramphenicol, gentamicin, and ciprofloxacin are commonly used antibacterial for this purpose. If the sample is taken from a sterile site, it is not necessary to use media containing antibacterial agents. 3. Plates are preferred because they provide better aeration of cultures. It has a larger surface area for better isolation of colonies, and greater ease of handling. Superficial mycoses are limited to the stratum corneum and essentially elicit no inflammation. This is usually a cosmetic concern, and does not cause discomfort to the patient. On the other hand, cutaneous infections involve the integument and its appendages, including hair and nails. Infection may involve the stratum corneum or deeper layers of the epidermis. Inflammation of the skin is elicited by the organism or its products. Superficial mycoses, again, is limited to the outermost layer of the skin. There are 4 types of infection under the superficial mycoses. 1. Pityriasis versicolor. 2. Tinea nigra. 3. Black Piedra, and 4. White Piedra. Malassezia species are members of a monophyletic genus of fungi normally found on human and animal skin. These lipid-dependent, commensal yeasts normally constitute greater than 80% of the total fungal population of human skin, and are frequently isolated in both healthy and diseased hosts. There are 14 currently recognized species of Malassezia, and few of the species are implicated as cause or contributors in several common dermatologic disorders such as pityriasis versicolor, seborrheic dermatitis, Malassezia folliculitis, atopic dermatitis, and psoriasis. Pityriasis versicolor is a chronic superficial infection of the stratum corneum. This is characterized by discrete, serpentine, hyper, or hypopigmented maculae. This is usually found on the chest, upper back, arms, or abdomen. These patches of discolored skin may enlarge and coalesce. Causative agents of majority of the cases are Malassezia globosa, Malassezia furfur, or Malassezia sympodialis. Lesions of pityriasis versicolor will fluoresce under Wood's lamp. The diagnosis, however, is confirmed by performing a direct KOH microscopic examination. Microscopic findings would show short unbranched, non-pigmented hyphae and spherical cells that are likened to spaghetti and meatballs. Pityriasis versicolor is treated with daily applications of selenium sulfide. Topical or oral azoles are also effective. It is important to let the patient know that the goal of treatment is not to eradicate Malassezia from the skin but to reduce the cutaneous population to commensal levels. Tinea nigra is a superficial mycosis of the stratum corneum. This is caused by Hortea werneckii, formerly known as Phoanolomyces werneckii. Formerly classified as Exophylla werneckii and Cladosporium werneckii. The infection is believed to occur as a result of inoculation from contamination sources such as soil, sewage, wood, or compost. The fungus exhibits lipophilic adhesion to human skin, and it receives nourishment from its use of decomposed lipids. It is characterized by the presence of hyperpigmented macules caused by accumulation of melanin-like substance in the fungus. The lesions are usually found in the palm and soles. It can also be found in other parts of the body, but the incidence is rare. Diagnosis can be done by microscopic examination of skin scrapings treated with 20% KOH. The microscopic examination will reveal thick, septate, branching hyphae that contain a dark pigment in their walls. Blastospores or chlamydoconidia may also be present. Culture of skin scrapings on Sabouraud agar at 25 degrees Celsius yields growth in approximately 1 week. Initially, yeast-like colonies that are shiny, black, and mucoid are present. Subsequently, the colonies become olivaceous brown and develop aerial mycelia in 2 to 3 weeks. Because tinea nigra is caused by a superficial fungal infection of the skin, topical medicines designed to eradicate the dermatomycosis are used. This includes keratolytic solutions like topical salicylic acid and tretinoin, topical imidazoles, topical pyridones, and topical allolamins. Black piedra consists of hard, darkly pigmented nodules of varying sizes that are firmly attached to the hair shaft. The most commonly affected area of the body is the scalp hair. Black piedra less frequently affects beards, mustaches, and the pubic hair. The fungus grows into the hair shaft. Ultimately, it may cause hair breakage because of structural instability. Diagnosis is made through KOH microscopic examination of the hair shaft nodule. Evaluation will reveal tightly packed and pigmented hyphae, ascus, and ascospores that are seen attached to the hair shaft. Shaving or cutting the hair is the treatment of choice. Topical antifungal and oral terbinafine can also be used for treatment of black piedra. White piedra consists of lightly pigmented, loosely attached nodules or gelatinous sheaths that have a soft texture. The most commonly affected areas of the body are beards, mustaches, pubic and axillary hair, and eyelashes and eyebrows. It may rarely appear on the scalp, where it can be extensive. Similar to the black piedra, the white piedra can also cause brittleness and breakage of hair. Diagnosis is also made through KOH microscopic examination of the hair shaft nodule. Evaluation will reveal loosely arranged hyphae, blastoconidia, and arthroconidia attached to the hair shaft. Shaving or cutting the hair is the treatment of choice. White piedra can also be treated by using topical antifungals. For both white and black piedra, when treated with antifungals, long-term monitoring is required because spontaneous remissions are common. Removal of hair is curative and requires no follow-up treatment. Recurrences are also rare. Dermatophytes, the most important and most common cause of cutaneous mycosis. These are a group of filamentous fungi that colonize and infect keratinized tissues, including the outermost layer of skin, hair, and nails. Unlike superficial mycoses, these infections evoke cellular immune response. The signs and symptoms of cutaneous mycoses vary depending on the infectious agent, location of the infection, and severity of the infection. The infection is called ringworm because it can cause an itchy, red, circular rash. Ringworm is also called tinea or dermatophytosis. These dermatophytes are restricted to the non-viable skin because most are unable to grow at 37 degrees Celsius, or in the presence of serum. There are a group of about 40 related fungi that causes dermatophytosis. And they belong to 3 genera. 1. Microsporum. 2. Trichophyton. And 3. Epidermophyton. Dermatophytoses are among the most prevalent infections in the world. Although they can be persistent and troublesome, they are rarely debilitating or life-threatening. The different types of infections are usually named for the location of the infection on the body. For instance, infection on the feet is called tinea pedis, commonly called athlete's foot. Infections in the groin, inner thighs, or buttocks is called tinea cruris, commonly called jock itch. Infection on the scalp is called tinea capitis. Infection on the beard is called tinea barbae. On the hands they are called tinea manum. And infection of the toenails, or fingernails, is called tinea unguium, also called onychomycosis. Other parts of the body such as arms or legs, tinea corporis. Because dermatophytes are acquired by contact with contaminated soil, or with infected animals or humans. The species can also be classified as geophilic, zoophilic, or anthropophilic depending on whether their usual habitat is soil, animals, or humans. Anthropophilic species cause the greatest number of human infections. They elicit relatively mild and chronic infections, and may be difficult to eradicate. Conversely, geophilic and zoophilic dermatophytes, being less adapted to human hosts, produce more acute inflammatory infections that tend to resolve more quickly. Trichophyton. These are dermatophytes that lives in the soil, people or pets. The genus involves anthropophilic, zoophilic, and geophilic species related to its natural habitats. Trichophyton species is one of the most important triggers of human hair, skin, and nail infections. Macroscopically, the trichophyton colonies tend to grow at a slow to moderately rapid pace. Texture is cottony or powdery in appearance. On the surface, the color appears to be white to bright yellowish beige or red-violet. While on the reverse side, the color appears pale, yellow, brown or reddish-brown. Its microscopic characteristics show septate, hyaline hyphae, conidiophores, microconidia, macroconidia, and arthroconidia. Chlamydospores may also be produced. Conidiophores are not well distinguished from the hyphae. The microconidia of trichophyton are one-celled and round or piriform in shape. They are numerous, and are solitary or arranged in clusters. The microconidia are often the predominant type of conidia produced by trichophyton. The macroconidia are produced in very few number. When identified, they are multicellular, smooth, thick-walled and cylindrical, clavate or cigar-shaped. Epidermophyton is a filamentous fungus, and one of the 3 fungal genera classified as dermatophytes. It is distributed worldwide. Man is the primary host of Epidermophyton flaccosum, the only species which is pathogenic. It often develops tinea pedis, tinea cruris, tinea corporis, and onychomycosis. Epidermophyton grows slowly, and it does not require any specific growth condition in culture. The colonies tend to create a dark yellow to greenish-brown or khaki-colored layer. Initially, the colonies appear flat, then it becomes elevated and folded in the middle. Epidermophyton microscopically reveals septate hyaline hyphae. The macroconidia is multicellular, smooth, and thin-walled. Its shape is clavate or club-shaped and appears in small clusters. Take note that Epidermophyton does not produce microconidia. Microsporum is a filamentous keratinophilic fungus included in the group of dermatophytes. It has the ability to degrade keratin, and thus can reside on skin and its appendages and remains non-invasive. Microsporum mostly infect the hair and skin, except for Microsporum persicolor, which does not infect hair. Nail infections are very rare with Microsporum species. Microsporum colonies are glabrous, downy, woolly or powdery. The growth on Saburod dextrose agar at 25 degrees Celsius may be slow or rapid, and the diameter of the colony varies between 1 to 9 centimeters after 7 days of incubation. The color of the colony varies depending on the species. It may be white to beige or yellow to cinnamon. From the reverse, it may be yellow to red-brown. Microsporum produce septate hyphae, microconidia, and macroconidia. The microconidia are unicellular, solitary, oval to clavate in shape, smooth, hyaline, and thin-walled. The macroconidia are hyaline, aconylate to roughened, thin to thick-walled, typically fusiform and multicellular. Here is a tabulated summary of some of the common dermatophytes, with their colonial appearance and microscopic morphology. Diagnosis of cutaneous mycoses, or dermatophytosis, is based upon 1. The infected anatomical site. 2. Type of lesion. 3. Examination with Wood's lamp. 4. KOH microscopic examination. And lastly 5. Culture of organism. The treatment for dermatophyte infection depends on location and severity of the infection. But generally, the treatment involves removal of infected and dead epithelial structures, followed by application of topical and/or oral antifungals. Topical therapy is recommended for a localized infection because dermatophytes rarely invade living tissues. Topical therapy should be applied to the lesion and at least 2 centimeters beyond this area once or twice a day for at least 2 weeks, depending on which agent is used. Then lastly, reinfection prevention. Examples of prevention would include keeping area dry, and avoid sources of infection, such as an infected pet or shared bathing facilities. Let us end this lecture with some key concepts on superficial and cutaneous mycoses. Superficial and cutaneous mycoses are among the most common of all communicable diseases. Most superficial and cutaneous fungal infections are caused by species of Malassezia, dermatophytes, or Candida. The growth of dermatophytes is inhibited by serum and body temperature, and these fungi rarely become invasive. Geophilic and zoophilic dermatophytes usually cause acute, inflammatory lesions that respond to topical treatment within weeks and rarely recur. Anthropophilic dermatophytes usually cause relatively mild, chronic lesions that may require months or years of treatment and frequently recur.